A new targeted treatment for cats with head and neck cancers may also help save human lives in the near future, says scientists. The first ever clinical trial of the therapy found that more than 1/3 of felines who received the treatment had their disease controlled with minimal side effects. The drug will likely be effective for humans with head and neck squamous cell carcinoma, HNSCC, which is notoriously deadly and difficult to treat, according to the American research team. There are two major findings from this study, says study senior author Professor Daniel Johnson. It showed us that it is possible to target a transcription factor that drives oncogenesis, which is something that has been notoriously difficult in the past. Also, it demonstrates that pets with cancer can be a good representation of human disease, and that clinical trials in pets may yield more reliable results than tests in mouse models. Professor Johnson says the drug is the first to target the transcription factor STAT3, which is present in a range of both solid and liquid cancerous tumors, including a majority of HNSCC cases. The idea to test the drug on pet cats arose when study first author Dr. Jennifer Grandis talked to her sister, who is a veterinarian. Oral cancer in pet cats, including HNSCC, are extremely difficult to treat, and most animals die within 2 to 3 months of diagnosis. One cat who benefited from trial was a 9-year-old black domestic shorthair named Jack. He was diagnosed with HNSCC, and vets gave him just 6 to 8 weeks to live. Owner Tina Thompson said, "It was just such a gut punch. We wanted more time with him. When I found out about this clinical trial, I knew I wanted him to be a part of it." Jack went to weekly treatments for 1 month, and during that time his symptoms and watery eye improved significantly. He ultimately lived more than 8 months beyond his most dire diagnosis. "Those 8 months were meaningful to us because he was there in our lives," recalled Tina. "Jack got to spend one more Christmas with us, and he loved our Christmas tree. He was worth every bit of the effort." None of the cats in the trial developed side effects that were attributable to the treatment other than mild anemia, according to the study published in the journal Cancer Cell. Of the 20 cats who were enrolled, 7 exhibited either a partial response or stable condition during the study period. When the research team looked at tumors and blood samples from the cats who underwent treatment, they saw that the compound was working in 2 ways. It not only blocked the activity of STAT3, but also raised the levels of PD-1, a protein associated with an immune response to cancer. Dr. Grandis said, "This study is a great example of how we can think more carefully about spending our very limited resources on studies in lab mice that are not even the best models of human cancers. By partnering with veterinary oncologists and doing clinical trials on companion animals, we can learn an enormous amount about how these drugs work while also helping people's pets." None of the cats in these trials were harmed, and many of them benefited. The researchers say that conducting clinical trials in pets can be a much better model of how drugs will work in humans compared to lab mice. The team is currently working with a small biotech company to advance the new compound in clinical trials for both pets and